Hey everyone, my name is Wang. I'm from San Diego, California, and my project is a computational application to identify significant changes in the 3D structure of DNA across multiple cell types. Work that'll help us understand the physical mechanisms behind everything from cellular differentiation to cancer. Today, I want to begin by briefly explaining what the 3D genome is, then by outlining an open problem in comparative genomics, before finally explaining how my approach fills that gap with a first-of-its-kind multivariate formulation. So, to begin, what is the 3D genome? Well, we can sort of envision it like this cord. If fully stretched, the DNA in a human cell would take up 6 feet. Inside a cellular nucleus, however, it is compacted and smushed into a space that is 1/10 the width of a human hair. Obviously, this isn't done arbitrarily. Instead, DNA adapts a complex 3D structure that acts as a global regulator of sorts, orchestrating transcription, methylation, and a range of other processes that guide how our genes function. One way that we can understand this structure is something called genome compartmentalization. Previous research has shown that the genome is spatially segregated into 2 compartments, the A compartment and the B compartment, which correspond to areas of active and inactive DNA. We can identify compartments by taking the first eigenvector of principal components analysis on a type of high-throughput genome mapping data called Hi-C. And in this resulting profile, areas with a positive sign represent the A compartment, and areas with a negative sign represent the B compartment. Compartment analysis is a very powerful tool. For instance, 12 to 36% of the genome changes compartment in cancer. And by looking at compartment changes in systems like these, we can find the genes and pathways that cause relevant changes in biological behavior. Unfortunately, as it stands, there are 2 main issues with comparative compartment analysis. Firstly, that we can only look for compartment flips. Because PCA values scale somewhat arbitrarily, the actual values in the compartment profiles don't tell us much about the A-ness and B-ness of a given compartment. As such, we only look for A to B and B to A changes. It is for that reason that number 2, compartment comparisons are only limited to pairwise settings. There is no way to systematically compare 3 or more cell types. As such, we are faced with a bit of an open question. How can we systematically compare compartment profiles from multiple cell lines? My project adopts a 2-pronged approach to solve this. By firstly, using multiple factor analysis, an extension of PCA that normalizes every dataset by its first eigenvalue, a number that can be thought of as a measure of the data's variance, before then performing a global PCA on all of the datasets for normalized compartment profiles. And secondly, rather than naively looking at compartment flips, it adapts a significance testing method that assigns p-values to every compartment bin based on how differential the compartment might be. It does this with a series of multivariate statistical scores, combining Mahalanobis distance, or a multivariate Gaussian, along with a biological replicate variability score in an independent hypothesis weighting, or multiple testing correction setting. I applied this methodology to several datasets, 2 of which I exhibit right here. The first of these is a gold standard, extensively characterized system of mouse neuronal differentiation, tracking the development from mouse embryonic stem cells to cortical neurons. In my results, I was able to recover existing confirmed compartment changes in literature, and was also able to exhibit biologically relevant examples of significant same compartment changes, in this case A to A, but also B to B, that correspond with levels of gene expression. This is a level of nuance that has never been achieved before. In human datasets, like comparison between breast cancer cells versus normal breast cells, I was able to do similar things, identifying important genes in cancerogenesis, and when performing gene set enrichment analysis on genes within differential compartments, my top results were hallmark breast cancer signaling pathways. I was able to achieve similar results in even larger experimental cohorts, with 10 and 11 cell lines in complex experimental hierarchies. And these results are available in a public preprint that is currently under review in a genetics journal. This entire application is freely available and documented on GitHub, and is currently being used by dozens of other researchers. With thousands of Hi-C datasets available and more being created every single day, this offers a new modality in comparative genomics that can help us identify the factors that drive development and disease.